Welcome to today's lesson. Today we are dealing with 2018 biology practical. Let's enter inside and see what it contains. In question 1, the photographs below represent 3 mammalian bones labeled E, F and G. You should be able to understand and to know the drawings of the various mammalian bones. In this question, they are not very visible, but we will still answer them. E was the scapula, F is the humerus, and G is the ulna and radius. Now you are told, with a reason, identify the bones. Bone E, you have said it's a scapula. Reason is because has glenoid cavity. This is the glenoid cavity. This one, the glenoid is the glenoid cavity. Yeah, that's the reason it has the glenoid cavity. Also has the acromion has acromion. This is the acromion, this one, acromion, and this is the meta acromion. Yes, this is the spine. Yeah, you should be able to know those parts. Those reasons are okay. In F is the humerus. F is the humerus because, as you can see, though it is not very visible, has a rounded head. Has a rounded head. But in E, the scapula, you should be able to know where it is found. It is found at the shoulder, here up at the shoulder. And the humerus, it's below the shoulder. The shoulder has the reason is has a rounded head that articulate with the glenoid cavity. Now this round-headed head is here, it articulates with the glenoid cavity. Now here, those two are from the arm. Another reason is has condyles. The condyles articulates with the radius and ulna. And G now is the ulna and radius. Ulna has, the reason is because they have a sigmoid notch. This is the sigmoid notch, this hole, the sigmoid notch. This is the ulna and the radius. Yeah, ulna and radius has the sigmoid notch. The reason is has sigmoid notch. Also they have the greater tuberosity and the biseptal groove. You should be able to know the drawings of the mammalian bone. But a question in this year, it seems that they can bring the bones, so students should be able to know all types and drawings. You should be able to relate various drawings and know how to label them. In B, you are told, name the joints formed at the anterior and the posterior ends of F. F is the humerus. This is the anterior anterior, and the posterior. Posterior is the lower end, here is up, this is lower, this is upper. Anterior and posterior, you should be able to know those terms, anterior and posterior. Now the joint in the anterior, the upper one, it is ball and socket joint. Ball and socket joint. And in the posterior, the lower end, is the hinge joint. They articulate there with the ulna and radius, the hinge joint. In C, state the types of movement facilitated by the joint at the anterior end of the specimen labeled F. We have seen the joint in the anterior end is the ball and socket joint. This joint, it rotates 360 degrees. So the movement is rotation of the arm. It rotates. Movement is rotation of the arm. It rotates in all directions, all planes, rotation of the arm. D, Roman 1. Name the substance found inside the living tissue of the specimen represented in photograph M. Photograph M, we have said, is humerus. Now in humerus, we found that it contains the bone marrow. Bone marrow, its function is helps in the manufacture of the red blood cells. Even we have been asked, state the function of the substance named. Help in the manufacture of red blood cells. C, Roman 1. Name the muscle bundle usually attached onto the front of the specimen represented into the photograph M. You are asked the muscle bundle of the humerus. You know that the muscle bundle in front is the bicep, while the muscle bundle that it is at the back, it is the triceps. Now you have been asked the front one, which is the biceps. Biceps. Spelling, you should be able to know the spelling. Biceps. State the function of the muscle bundle. The function is it relax and contract to move the lower arm. Contract or relax to move the lower arm. Question 2. Below is a photograph of the blood cells smear from a normal individual. The arrangement is arbitrary and the number of blood elements is greater than that would normally occur in an actual microscope field. Although they are not very visible, but a student should be able to know the drawings of all the blood vessels. Like example, in red blood cell, you should be able to know they are biconcave in shape. In white blood cell, you should be able to know that they engulf themselves. They engulf or surround. They have the amoebic movement. Now, J, name the blood elements labeled J, K, and L. J we see is very small. It is very small, but it is a cone shape. It is a biconcave in shape. Now it is the red blood cells or the erythrocytes. And then K, as you can see, they have the amoebic movement. They resemble amoebic movement. Automatically, this is the leukocytes. sites or the white blood cells. They are very small. They are very small, as you can see, but they are the thrombocytes. Thrombocytes or the platelets. State one function of each element named in a Roman 1 above. The function of the red blood cell help in the transportation. These help in transportation of oxygen and carbon dioxide. And K, which is the white blood cell, they protect by engulfing. To engulf is to surround. Engulfing pathogens. These are the microorganisms causing diseases. Pathogens. L helps in blood clotting, which is the platelets help in blood clotting process. Yeah. In B, the photograph below is a section of a human intestine of a patient suffering from a common parasitic disease. Even though it is not very visible, this is a disease that occurs in a stomach. Now, there are very many diseases, but these ones are caused by parasites. Now, the disease is amoebic dysentery. This is the amoebic dysentery. It occurs in the stomach. Name the parasite that causes the disease. This simply are asked the causative agent. Now you know that the causative agent is Entamoeba histolytica. You should know this is a scientific name, so you should follow the rules, writing the scientific name by underlining them separately and starting with the generic name. You should start with the capital letter, with the species name or specific name with small letter. State 2 control measures for the disease. You should boil water before drinking. Boiling of water before drinking. Also, washing hands or fruits. Washing fruits or hands before eating. To prevent the amoebic dysentery. State 2 effects of having the part labeled G in patient's intestine. Part labeled G, these are the wounds in the stomach. The wounds, the effect is that you'll have pain, especially during feeding. You'll have pain especially during feeding. You have pain. Also, there will be blood stool. Even impaired absorption of digested food material. There will be impaired digestion or absorption of digested food material.
I hope you are enjoying the lesson. Do me a favor and subscribe. Thank you.
In question 3, you are provided with specimen labeled H. With the aid of hand lens, examine the external feature of the specimen. The specimen H was the bean pod. A pod from a bean. H was a bean pod. Bean pod. Now you are required to examine using the hand lens the features. A, you have been asked, what part of a plant is specimen H? Now the bean pod. You say this is a fruit. This is a fruit. Give two reasons for your answer. Why is it a fruit? Because it has two scars. That's the first reason you should give if you have been, as if you have written that your answer is fruit. Has two scars. That is the point of attachment. The receptacle has two scars. There is presence of seeds. Now presence of seeds and etc. Now you have been told, open up specimen H longitudinally. Longitudinally is, you know how longitudinally looks like. Use a hand lens to observe the internal structures of specimen H. Yeah. Now you are being told, draw and label the internal cut surface and associated structures of specimen H. Now you are required to draw well-labeled diagram of the bean pod which you have opened it. I should be able to know the drawing. Next, explain how you will determine the magnification of drawing made in B Roman 1 above. Now you should be able to know linear magnification. That is when you are using hand lens. Linear magnification is equal to the length of drawing all over the actual length of the object. Actual length. That is the linear magnification. Roman 3. State the mode of dispersal for seeds of specimen H. What the mode of dispersal? This is simply the self-dispersal mechanisms of the self-explosive mechanism. Self-explosive mechanism. Self-explosive mechanism. Explain how seeds of specimen H are dispersed through the mode stated in B Roman 1 above. The dry pods, due to water loss from the pod and pressure within the pod opens through the lines of weakness. Then the seeds are thrown away. Now you should say the dry pods loses water and a pressure is created, within the pod. Then the pod opens violently along lines of weakness. Then the seeds are thrown away, or they are dispersed from the parent plant. Thank you. That's over.